Thank you. Yes, so I would like to talk to you today about a project which is actually very long historic roots at the Lund University, where researchers have been working on developing a stem cell therapy for Parkinson's disease for more than 30, 40 years. And I think today actually we've reached a point where we're very close, probably actually a lot closer than people would have thought 5 years ago. And right now we're actually in the middle of GMP translation and planning all the preclinical data for actually getting regulatory approval to go into patients. So I think this is a very exciting time to be in stem cell research. So I'd like to talk to you about some of the hurdles in taking a research project and bringing it to the clinic, and how we've gone about to solving these problems. So just a brief introduction about Parkinson's disease. So Parkinson's disease is a neurological disorder. The hallmark of the disease, which most people know of, is this typical tremor, typically of the hand. But this is actually not the main problem of the patients. The main problem of the patients is a very pronounced rigidity. So they have a hard time actually initiating movements of their muscles. They can think that they would like to pick up their cup of coffee, but in fact they cannot actually make their muscles execute that order. So sometimes they'll explain it as they feel trapped in their own body. They can think they want to do things, but they cannot make their body actually do it. And they say this is extremely, extremely frustrating. Also, they have a tendency to fall often because they have this postural instability where they kind of have a gait where they have a tendency to fall forward. The main reason for these motoric symptoms of Parkinson's disease is the loss of a very specific nucleus of neurons, of dopaminergic neurons in this nucleus called substantia nigra. And the reason it's called substantia nigra is because it actually is black. And this is not stained. This is what it looks like just when taking the brain out of the patient, because these neurons are actually pigmented. So you can see this is a controlled subject where you see the dopaminergic neurons lying right here. And this is now a Parkinson's disease patient in the terminal stage where there's pretty much no neurons left. And you can see also on this graph, as the patient progresses, after 20 to 30 years, there's less than 10% of dopaminergic neurons left in the brain, and it really only goes in one direction. Today, what we do is we treat these patients with dopamine-enhancing pharmaceuticals to boost the release of dopamine from the neurons that are left in the brain. There's also neurotechniques like electrical deep brain stimulation, which I'm not going to talk more about. But the problem of all of these therapies that we have today is that they don't change the course of the disease. We have no disease-modifying treatment. The disease is, no matter what we do, it's going to go in this direction. And this is a problem because the treatments that we have actually work really well in the beginning of the disease. So this is L-DOPA, which is this was discovered in the 50s, and we're still using it today. It's actually still the main therapy for Parkinson's disease patients because it works really well, at least in the beginning. So what you see here is that when the patient plasma concentration comes below this line, the patient has no benefit of the treatment. And when the plasma concentration comes above this line, the patient gets side effects, which is dyskinesias, or involuntary movement. So the patient will be having a lot of movement of the arms and legs, which they cannot control. So when we're in here, in this area here, this is the therapeutic window where the patient has a good effect of the treatment without having side effects. And this is now the patient will take a pill, the concentration will go up and it will drop, the patient will take a new pill, it'll go up and down, and so forth. So you see here in the beginning of the disease, you have a very nice therapeutic window. As the disease progresses, the therapeutic window becomes really, really small. And now the patient only has either no effect or side effects of the treatment. And this is because there's basically no dopaminergic neurons left in the brain to buffer the capacity of L-DOPA. So now L-DOPA is taken up by other neurons, and then you get the side effects of the treatment. And at this stage in the disease, we really have very little to offer the patients because we've already exhausted all the dopaminergic therapies that we have. So this is why this hypothesis emerged that because Parkinson's disease is caused by a very localized loss of a very specific type of neurons, can we actually replace those neurons with new neurons? And this is work that's been ongoing at the University of Lund actually since the 60s and 70s, led on by these two professors who are still active actually in Lund. They pioneered this whole field. And their hypothesis was that, well, could we take tissue from the human fetus, the exact tissue of the human fetus that gives rise to these dopaminergic neurons, put it in suspension and graft it into the patients. And actually the first transplantations of this kind were done in Lund already in 1987. And over the years, I think between 20 and 25 patients have been grafted in Lund. And also patients have been grafted in other places of the world after seeing these first transplants that were performed in Lund. And here I'm just going to show you a video of what this can look like. This is a patient who was grafted in Halifax in the US. Here you see what the patient looks like before transplantation, a typical symptoms of Parkinson's disease. She has a hard time walking. Her steps are very small because she cannot initiate large movements. And basically the whole upper part of her body is very rigid. There's no swinging of the hands or anything like that. And she walks extremely, extremely slowly. You can also see that she's extremely thin. So this is a very prominent hallmark of Parkinson's disease, that they lose weight. This is now the same patient 5 years after transplantation of fetal tissue. And you can now see how the walking of the patient is much more eased. There's hand swinging, which is something you would never see here. And basically you can see there's a major improvement of the patient and an increase in weight. And for someone who works in the field of Parkinson's disease, this is something you would never see. This would never be a natural course of the disease. The disease basically only goes in one direction, and that's deterioration. So we know from stories like this, individual patients like this, and there's also quite a lot of examples in Lund, although we don't have the videos from them, that the transplantation can really work excellently. We've had patients who've been off medication for more than 16 years. We've just last month, we got a brain from a patient who had been transplanted 24 years ago, and the transplant still looked beautiful and innervated the whole striatum, which is the part of the brain of dopaminergic innervation. So we know that this therapy can really work, and we also know that it works through what we think it does, namely that it re-innervates the dopaminergic pathway of the brain. So this is a scanning of a patient before transplantation. This is a PET scan that looks for dopamine activity in the brain. And here is the same patient after transplantation, where you see an increase in the dopamine signal. So this is really the key to the success of these experiments, the acquisition of aborted fetuses. And we know that this is a great therapy, so why don't we just transplant Parkinson's disease patients, because we know that it works excellently. The problem, of course, here is that it's extremely, extremely hard to get enough tissue to transplant many patients. So for one patient, we actually need between 6 to 8 fetuses. They need to be good quality, they need to be the right age, they need to be fresh. We cannot store them or store the tissue. So actually right now in Lund, we're part of this EU project where we're trying to reproduce these results under a more controlled environment, where we want to transplant 20 patients in collaboration with the University of Cambridge. And this project we were supposed to transplant the first patient 2 years ago, and the first patient was actually transplanted 2 weeks ago. And this is because it's just so difficult logistically to actually get this to work, to get the tissue, to get enough tissue, to get enough good quality, to have the patients ready, and so on. So even though we know that this works, this is not the future. We really need to get these cells from somewhere else. So of course, we, as many others, are taking this strategy of producing the cells in the lab, and we are then focusing on producing them from human embryonic stem cells because this is really a cell type where we know that we can get the true type of neurons that we want. And in order to do this, we need to understand how these neurons are formed in the embryo, and we know that the neurons are derived from this specific part of the brain, which is the ventral midbrain, and we need to be able to mimic this process in the lab. So we've worked on this for some years, and actually, if you break down the patterning of the early human brain, you can simplify it by patterning through a few series of gradients. So this is a schematic of the early neural tube, and we know that there is a gradient of Wints from anterior to posterior with increasing concentration of Wints in the posterior part of the brain. We also know that patterning from dorsal to ventral involves the activity of BMPs from the roof plate and the activity of sonic hedgehog from the floor plate. And this now then causes the formation of these regions in the human embryo. So we've seen that we can actually mimic the activities of these gradients in the lab. So if we look at Wnt signaling, it's actually very hard to work with Wnt in the lab because the biological activity of commercially available Wnt is quite low. But we can activate Wnt chemically by adding a GSK3 inhibitor, which then intracellularly activates the Wnt pathway. And what we've shown is that if we now differentiate human embryonic stem cells towards neural cells, and we change the concentration of just this single compound in order to change the activity of Wnt, we can control where we are in the brain. So at low concentrations of the compound, we get forebrain, so the anterior part of the brain. At medium concentrations, we get midbrain, and at high concentrations, we get hindbrain here. And this is just by altering the concentration of one single compound. And we've then tested these neural progenitors to see if they really are authentic from different parts of the brain. And what we've seen is that if we pattern them for only 10 days in vitro, so we differentiate them, they start by ES cells, we differentiate them for 10 days under these different concentrations, and then we transplant them to the brain of the adult rat. And what we see here is that this is now a Parkinson's disease model where we've eliminated all endogenous dopaminergic neurons. And the dopaminergic neurons is what we stain for here, this marker TH. So here you see in the midbrain conditions, which is the part of the brain that would normally give rise to dopaminergic neurons, we see that even though we only pattern them for 10 days in vitro, now they really do give rise to dopaminergic neurons, whereas the progenitors from other part of the brain, even though they were put into the same environment, these do not give rise to the midbrain dopaminergic neurons. So actually 10 days in vitro is enough for the cells to be completely regionalized into their specific fates, and they keep these fates even after transplantation. We can then test the activity of these neurons from a Parkinson's disease point of view. So we have this model where we lesion the dopaminergic system in only one side of the animal's brain. And then what happens is that when we stimulate dopamine activity through giving the rat amphetamine, now the rat will start rotating in one direction because there's only dopamine release in one side of the brain. It looks like this. It'll start rotating around and around in the dish, and we can then quantify how many times it rotates. And then we can see it rotates so many times per minute. Then we can add our transplant, which is patterned towards ventral midbrain, here VM. And we can see over time as the cells mature, the rat will now stop rotating, and now it'll actually start rotating in the other direction if we add too much dopaminergic neurons. Now, if we instead pattern the cells towards the forebrain, which is the gray here, then we don't get any dopaminergic neurons, and we also don't get any reversal of rotations. So we can see that these cells can actually reverse motor behavior in this Parkinson's disease model. We can also look at limb use. So this is a model where the rat is put in a glass cylinder, and then because it's curious, it'll pop up and see what's around it. And when it's lesioned in one side of the brain, it'll only use the contralateral paw. And then when we transplant the cells into the brain, it'll now start using both paws. So we can actually reverse motor behavior in these rats. So there's been a lot of criticism that, well, some people say it's very easy to cure a rat or a mouse from Parkinson's disease because you lesion the brain, and you add a blob of cells, and it's easy for these cells to really take up the whole volume of the brain, and just by the fact that the volume of the brain is so small, it's easy for them. You don't need a lot of cells to actually reverse the behavior. So in order to challenge the system, because this is true, because in the human brain, the volume is much larger. You need the cells to actually activate a large volume of the brain. So in order to challenge the system, what we did was, we would normally transplant the cells into here. This is striatum. This is the target site of the neurons. So the neurons are supposed to innervate this part of the brain. But in fact, they are normally located not here, but down here, which is more than a centimeter away from the actual target site. So we challenged the system. So here's what they're normally located. We challenged the system, and then we put the cells, instead of putting them here, we transplanted them here into the substantia nigra, their endogenous site. And we saw this is now fetal tissue, so tissue obtained from aborted fetuses. And we could see now that this tissue could actually grow all the way up to the striatum and even all the way up to the cortex. And we tested the same with the embryonic stem cells, and we saw the same thing. The cells could actually grow their axons all the way out to the prefrontal cortex and the striatum, which are normal target innervation sites for dopaminergic neurons. So we know now that the cells can innervate more than a centimeter away from where they're put, and this is clinically relevant. This means that, in theory, they can innervate the whole human striatum of a Parkinson's disease patient. So we're at a point now where we're thinking, well, these cells are actually pretty good. We feel like we have something to work with. We could go into Parkinson's disease patients. So we got a grant from the EU to actually do this. It's called neurostem cell repair. It's under FP7. It's a €6 million funding for 4 years. And it involves collaboration with this company, Roslin Cells in Edinburgh, who has a GMP facility for embryonic stem cells. So they are supposed to be the ones who will produce the cells for the clinic. So what we need to do is we need to adapt the protocol to make a ready-to-use product, and here we want to make a cryopreserved ready-to-use product. And we need to explore the possibilities of maybe having a purification step, because this could be required from the authorities. And then we hope we'll be able to produce a final GMP product in 2017. So what do we need to do in order to go to the clinic? Well, we need, of course, to GMP adapt our protocol. So we need a GMP cell line, we need a reproducible and reliable protocol, we need animal-free components, GMP standard reagents, and a possibility of cell sorting in case this is required from the authorities. We need to be able to predict the in vivo efficacy of our cells, so we know that one batch will not get a completely different in vivo efficacy than another batch. We need to make standardized quality control for our cells, so we know that we have the same product every time we produce it. And finally, we need to be able to freeze our cells so we can have a ready-to-use product and to ship it into clinics without too much variation. So I'm just going to show you some of these points we've gone through. First point, which is really critical, because this is the first thing you need to do, but it's actually not so simple, is to choose a candidate GMP cell line. And there aren't so many yet to choose from. There's a lot of work ongoing. We'll get a lot of new cell lines soon, I think. But at the moment, there's not so many. In the EU, pretty much all of our GMP-grade cell lines are located in the UK. And since we're collaborating here with Roslin Cells, we are using cell lines from Roslin Cell Company. And this is just to show you that it's really important that we have a large library of GMP cell lines to choose from. Because even though we're very good at making cell lines, we're very good at characterizing them, we're very good at making it with small variations and so on, still, for some reason, each single cell line has its own properties when you start differentiating it, and you cannot predict this. So all you need to do is just test them and then pick the one that works the best for your property. And here's just an example of 3 different cell lines where we differentiated them towards our target population of the ventral midbrain. What we see really is huge variations in the yield. So these 2 cell lines were very heterogeneous. whereas this cell line down here generated more than 90% purity of our target population. So of course we chose to go with this one. But this was pure luck, right? We might as well have had 3 cell lines that didn't work very well. So we're using this cell line now to perform all of our GMP adaptations. And we also need to adjust our conditions during the pluripotency, maintenance of pluripotency of the cells. We tested a bunch of different combinations of matrices and media, and really what we see is that there's large variation in the morphology of the cells, although they seem to be pluripotent, all of them. There's also variation in how fast they grow. So they definitely grow faster on laminin 521, and what we see is that from all the conditions that we screened is that this condition here, laminin 521, in combination with IPS brew from Miltony, is absolutely the best condition. This is the only condition where we've never seen our cells at some point suddenly crash or at some point suddenly start becoming flat and grow slower. Strange things happen, which we've seen with E8 and vitronectin and StemPro. So we've decided to stick to this condition also because this is GMP compatible. And actually in this condition, we can split our cells 1 to 100 every 7 days very reliably, even across different cell lines. And I think this works very nicely. And I have to say, these are actually karyotypically normal. We've tested that, and there's no abnormality in them. So then we need to adapt our differentiation protocol. And the original protocol was based on an embryoid body step. And this was because we really didn't have any good matrices to plate our cells on, which was GMP compatible. At the time we made this protocol, the only matrix we had was Matrigel, which we know in EU would not be approved. So we decided to go with an embryoid body step here with the first 4 days, and then we plated it on polyornithine, fibronectin, and laminin. And then on day 16, we would transplant our cells. And here we would have factors for neuralising the cells and factors for patterning the cells. And this protocol, we were very happy with it because it generated a very high yield of our progenitor cells, over 90% purity. But the problem was that there was actually quite a lot of variability in the yield of the cells. And also when we now tested our GMP cell line, suddenly the yield was even lower than we used our H9 cell line. And finally, our GMP facilities in Roslin cells were not at all happy with this EB protocol. They thought it was not so easy to standardize, and they would much rather have an adherent protocol. So we went back into the lab to see if we could adjust the protocol for, instead of having an embryoid body step here, if we could just have one step here with having the cells adhered to a matrix. And of course here we had to take into account that Matrigel was not a good choice because it's undefined animal-derived product, and also laminins derived from mouse sarcoma would also not be suitable for GMP-compatible production. So we were looking for a matrix which should be recombinant and should support specifically neural differentiation and not so much differentiation towards other lineages. And here we're in contact with Biolamina, of course, and we tested their panel of different laminins. And actually what we saw was that on day 1, the cells would adhere to pretty much all the laminins. But then the cells were growing, we saw large variations in the morphology of the cells. Here, for instance, they do not at all look neural here, and they were very heterogeneous on the plate. From these experiments, we saw that the growth was best on laminin 111, laminin 421, 511, and 521. So since 511 and 521 also support growth of pluripotent cells, we wanted to avoid this because this was a matrix we wanted to use for differentiation, and we did not want a matrix which would also support pluripotent cells. So we kind of sorted those out. And since laminin 521, from a biological perspective, did not seem so relevant because it wasn't really expressed in the brain, it was expressed in tumors and non-neural tissue, But then laminin 111, on the other hand, is expressed in the developing brain. So from a biological perspective, this seemed like the most relevant choice to make. So we decided to continue with laminin 111. And what we really like about this matrix is that it does not support growth of pluripotent cells. So if we plate the cells, the day after plating, we really see that colonies can adhere both to laminin 521 and to laminin 111. But if we now keep them under pluripotency conditions here in IPS brew, on day 4 already, this is hard to see in this picture, this is now a confluent layer of cells. So here the cells are already taking up the whole well, and here the cells has formed kind of spheres or balls of cells. And now if I take this plate and I change the medium, they'd all be floating in the plate. So pretty much on day 7 there was no colonies left. So if I tried to plate the cells on laminin 111 under pluripotency conditions, they would not adhere at all. And we could also quantify this in Matrigel 521 laminin 111. But on the other hand, if I took the same cells, plated them on the same plate, and now put them in differentiation medium instead of pluripotency medium, now they would adhere and they would form a very nice cell layer on day 7. So it seemed that this matrix would really support the differentiation towards neural tissues, but would not support pluripotent cells. And this is exactly what we were looking for our protocol. A couple of other adaptations we did was we removed B27 from the medium because there is no GMP standard B27 at the moment. And also this was to reduce batch-to-batch variation of the differentiation. And at the moment, we're trying to substitute all our reagents in the protocol with GMP-compatible reagents from Milteney. And this is, of course, the long and tedious project where everything has to be tested one by one in different concentrations. And right now, we're at a point where when we look at even just the first phase of the protocol, from switching from an embryoid body protocol into a laminin 111 protocol, we could increase yield 11 times just on the first 11 days of differentiations. So we could start with 10,000 cells, and now on day 11 we would have 1.4 million cells. So the yield really went through the roof just by changing the protocol with the matrix. And right now, this is now the version of the protocol we're working with right now. This was the original embryoid body protocol. And this is now the GMP protocol. We have the colonies on laminin 111. We took away B27. We added FGF8 here. And overall, when we look at the cell yield now in the new GMP protocol, we got a 30 times increase in cell yield on day 16 compared to the old embryoid body protocol. And in fact, what this means is that if we start with a 6-well plate of embryonic stem cells, which is around 14 million cells in our hands, we can, by day 16, which is the time when we transplant the cells, produce 2.5 billion neural progenitors for transplantation. And this is, in fact, enough for treating 500 patients at our current estimates. So we can actually start with very few cells and get enough cells for many hundreds of patients. So we think this is very encouraging. We think that this now means that we're actually ready to scale up production for our first clinical trial. We've done some preliminary studies of testing the GMP cell line in vivo. It looks encouraging. We get survival of the cells. There's no tumors or any overgrowth. The cells form neurons with large, nice outgrowth into the host tissue, and they're very dense and dopaminergic neurons. And we've just gotten the first results actually yesterday from transplantations with the GMP protocol on laminin 111 with our GMP cell line, and we've seen functional recovery in the Parkinson's disease to rats. So we're very encouraged by this. So this is where we came to in the GMP adaptation. We're using the cell line, we're using laminin 111, we're using reagents from Milteney and Biolamina for GMP standards. And then we're trying to introduce a step of cell sorting. And here we're collaborating with Milteney in Germany. So I don't know how many of you are familiar with Milteney, but they have this this technology where they can sort cells based on magnetic beads, or magnetic, it's called magnetic cell sorting. So magnetic antibodies. And they've developed for us a marker which recognizes specifically our target population. So this is the staining for the cell surface marker, and this is our target population expressing LMAX1A, FOXA2, and there's a very nice overlap here. And we can now pull these out with the max sorting, and we can see that the purified population is much more homogeneous than the non-purified. And in this experiment, we used, to verify the use of this purification step, we used an iPS cell, which actually turned out to be very dirty. So when we transplant this cell line, we get, which we never get with embryonic stem cells. With these iPS cells, we get huge tumors, and they look kind of nasty with a lot of even non-neural tissue in between. So this was a good model to really test the potency of our cell sorting step. So when we take these cells and we sort them for this marker, which, I'm sorry, I can't give you the identity because I don't even know what it is. This was developed in Milteney. We can sort out the cells and we can now get very nice graphs with neural identity and TH cells. So this is kind of for us a backup in case we're asked to sort the cells, because we are grafting the cells at a very early stage, so it might be required from authorities that we sort them. So we're now testing this in our ES cell line. So then we're also working on making vivo efficacy more reliable. We're doing this by really going back. We've grafted over 600 animals now in the lab. We're going back now to see, is there any correlation between the cells we grafted, the RNA expression in the cells we grafted, and the actual outcome of the graft 6 months later? Can we find markers which can predict whether this cell batch will become a good graft or a bad graft? And then finally, we're working also on cryopreservation. We've tested a lot of different cryopreservation cocktails, and we've ended up actually using one that we made ourselves, which was just adding 10% DMSO to the cell culture medium. The cells look the same before and after. They express the same markers before and after cryopreservation. And we can even, we grafted them in 2 separate experiments where we see they're a little bit smaller. So in this experiment, we really took the cells, as in a clinical setting, we took them directly from the freezer, washed them twice by centrifugation, and then transplanted them into the brain. So this is to mimic what would happen in the clinic, that we ship a frozen vial to the clinic, and they would take it, wash it, and transplant it into the brain. And here we see the cells survive very nicely. The graft's a little bit smaller, so we probably need to transplant a little bit more cells. But the potency of the cells to generate dopaminergic neurons is exactly the same. So we're right now at a point where we need to address these 2 last points, but this is underway. And I think we're moving steadily towards the clinic. We've already addressed most of our GMP points. And our final timeline looks like this, that we would like to train GMP personnel by the beginning of next year, produce the final product by early 2017, perform preclinical safety and efficacy. Of course, here, because they're ES cells, we'll probably be required to do long-term teratoma studies in mice, and then apply for clinical authorization. So the production here, our plan is to produce 1 batch of cells of 300 frozen vials with 5 million cells per vial. And this batch would then be enough to perform the whole quality control, all preclinical safety and efficacy testing, as well as the clinical phase 1/2. And the reason we want to do this is because then we can really validate the product that we're actually putting into patients. So we know that these cells have been tested in animals. And from our current estimates, we could produce this whole batch starting from just 4 wells of a 6-well plate for differentiation. So this is the group involved, a group of Malin, Parmer, and Lund. And then this is part of an EU project, the neurostem cell repair, with also people from Roslin cells and Milteney involved. Thank you very much.